Ah, entonces no se ve. Sí, toda la luz. Yo vine la semana pasada y todo bien, pero en la química no me lo prestaron. Me dijeron que no.
Laura, ya estamos en
Ah, ya
transmisión.
estamos en vivo. ¿Y se ve la presentación para ver? Yo sí.
Sí, tenés que compartir la pantalla.
Ah, sí, perdón, es que a veces me enredo con esto del Zoom. Tienes que habilitarme.
Yo. Esto está bien curioso. Listo. Y en principio yo me podría conectar a la cámara para cambiarle la posición, o eso ya sería. Sí.
Ahora sí, ¿verdad? Entonces, si le sigamos, Gianluca quiere poner esta a la par, entonces ve las dos
Mira, lo que estoy
simultáneamente.
viendo es.
En este momento estamos en vivo, pero eso se puede detener.
Aquí estoy viendo esto, la cámara larga, pero en la
Ok.
traducción, bueno, parece que esto.
Doña Carmen, todos son conocidos, no se preocupe.
Pues,
Podemos hacerlo con
Laura.
ellos ahí.
Así se está transmitiendo.
Ok, perfecto, qué dicho. Tal vez quítenle las caritas a los que no tienen video para que solo se vea. ¿Qué pasó, mi amor? Ya vamos a comenzar.
Si quieres, yo me muevo para. Tenemos que hacer mayor cosa. Sí. ¿Tienes opinión pequeña? Sí.
Ahora está prendido este micrófono y ese parlante. Cuando terminemos hay que apagar este micrófono y prender eso. Entonces ya de nuestra parte, ya estamos al video. Le podemos poner pausa un momentito que se detenga. Ah, bueno, sí, o mute. O nada más el sonido, entonces.
Hi, Gianluca. Can you hear me?
I can hear you, Theo.
Okay, great. Now we are about to start, but Carmen Madrid, she is the president, and she will give the rules of the doctoral examination.
Okay, perfect.
Es el máximo, es el volumen máximo que tengo, pero bueno.
Good morning. I want to thank all of you for attending this appointment today. The final examination of the student Laura Rojas. The name of the project is development of a biomechanical stimulation study system for bone-like and muscular scaffold. The members of the committee, as you know, is Dr. Teodolito Guillén, who is the advisor, Dr. Ricardo Starbeck Perez, Dr. Leonardo Leser Rojas, and Dr. Gianluca Tosi, who is in internet right now. And my name, Carmen Elena Madrid, who presents the evaluation process. First, I want to explain how is the dynamic of this evaluation. Basically, we have two different stages. The first stage is the evaluation of the thesis that you already sent the grades. The average of this grade is 94.1. Even though I need to ask you if you agree that she can continue with the second phase, that is the evaluation of the oral exam. All of you agree?
Yes, yeah, I agree.
Yes.
Thank you. After that, she's going to have 45 minutes to make the presentation, and also different public is going to join us in this presentation. Some of the public are present here in the room, and others are for the connection of the internet connection. After that, all the public will leave the room, and we are going to start the phase of the question, answer and questions. Every of you is going to have 20 minutes in one first round, and then one second round with 5 minutes. That could be a question, a final recommendation. When we finish the question and answer, we are going to have a deliberation or a delivery of the final grade for her and decide if she approved or not the examination. All of you understand?
Yeah.
And also I want to ask if you received the evaluation rubric. All of you have it?
Yes. Yes.
Okay,
I think the important is Gianluca, but it doesn't have it here printed.
okay.
But do you receive the Gianluca?
Yes, I have it.
Okay, great.
Okay, we need to sign it at the end.
No, porque tengo.
The doctoral exam rubric have some guide numbers that you can use as a reference, the 60, 80, 90, but you can assign a 95 or 98. It's just a reference. When we finish this part, I need that you send me this paper with your signature and the average of your grade. After that, we are going to read the final decision. The exam is going to be closed. Everything is clear? Okay.
Yep.
So now we are going
Do
to,
you have some question, Gianluca, for the examination process?
No, it's fine. I'm I sort of understood everything and the process. Yeah. So yeah, that's fine.
okay. We are going to call all the people.
Este. Bueno, entonces ya estamos. Este micrófono se escucha, ¿sí? Y ese parlante. Thank you.
Good morning, everybody. In the room, I'll also information of the website. In the name of the doctoral program, we are going to open the final exam. Student Laura Rojas. The title of the project is Development of a Biomechanical Stimulation Study System for Bone-like and Muscular Scaffolds. Laura is going to have 45 minutes to make the presentation. Go ahead with your 40 minutes.
Okay. Thank you for the introduction. So welcome everybody here and also in the online presentation. I'm very happy that we are all together today. I will start with the title of this project, that is Development of a Biomechanical Stimulation System, Study System for Cellular Growth in Bone-like and Muscular Scaffolds. To begin with, I will start with how this presentation is divided. I will talk first of the background, then the motivation, then the main goal, followed by the methodology, results, and discussion, then the contributions of the project, conclusions, and future work. So to begin with, I would like to give you a bit of background of the project. In this project, we will be referring mainly to two tissues, which are trabecular bone and skeletal muscle. These both tissues are related with shape and locomotion through the body, and these are live and can regenerate by themselves. Since these tissues are live and dynamic, they can suffer some diseases. For example, skeletal muscle can suffer from dystrophies, and in case of trabecular bone, there is an age-related disease called osteoporosis, in which bone mass is lost through aging. Moreover, these two tissues can also experience trauma or injuries. When injuries are small, these two tissues can regenerate by themselves. However, when the injury is large or a large defect is present, they are not able to repair themselves. The therapeutic approach for treating this type of trauma is by using grafts or artificial implants. However, artificial implants lack of chemical factors on their surface that may initiate cell migration towards the lesion. Therefore, there is still a demand for novel biomaterials to be used as cell colonized implants. Two key elements that can be used as that are necessary for cell colonized implants are the scaffolds, which should mimic the properties of the tissue that we want to replace. And also, as I said before, they should be cell colonized. In order to achieve a cell colonized scaffold, we need a bioreactor. A bioreactor provides means to control physiological conditions and also to change external factors. What are external factors? External factors can be chemical, biological, or mechanical, and can induce a certain response in the cells that we are working with. So keeping all of this in mind, I will present to you the project's main goal, which is to develop a mechanical stimulation in vitro test system for cell-seeded scaffolds that mimic skeletal and bone tissue. Now I will show you the specific objectives of the project as some diagrams. So first, we wanted to develop or to prepare some scaffolds using biopolymers in literature, you may find scaffolds of other materials. However, we wanted them to be biocompatible. And as I said in the beginning, scaffolds will be representing or will be done trying to mimic bone and muscle tissue. In addition, we wanted to design and fabricate a mobile in vitro system. What I mean by mobile is that it should be portable and self-sufficient, so you can move it from one place to another. And this in vitro system has to have the characteristics of a bioreactor. Therefore, it has to control physiological conditions, and also, as it says in the main goal, it has to transmit mechanical stimulation. When dealing with anything that has to do with cells, we have to consider also sterilization, because everything has to be sterile beforehand, and not all sterilization methods are proper for all scaffolds or for the system. So we had to do also a research to establish which sterilization procedure we were going to be used. And finally, we had to do the test of the in vitro system in a universal testing machine that is the one in charge of making the loads on the scaffolds. Now I will go into the methodology. I have to clarify that I will go through the methodology briefly due to time constraints in this talk. And if I have to go through some details, I will address them through the results. The methodology, I want to explain it as a timeline so that everyone can understand what we did and how it was done. So to begin with, we started with scaffold fabrication and characterization. 2 sets of scaffolds were fabricated and characterized. They were independent from each other, one for muscle, one for bone. And also the characterization of their mechanical properties was made using several tests. And in addition, some cell viability studies were done to see if the scaffolds were toxic or not, and if they support cell anchorage on their surface. Now, working in parallel, we did the design and the fabrication of the in vitro test system. These had to be done hand in hand because the scaffold fabrication, the scaffold sizes placed constraints over the in vitro test system. So we have to consider the size of the scaffold, the mechanical loads that were going to be used. So we did that in parallel until we have the final design. Also, we chose to use biologically inert material so that we can do it so that it could work as a bioreactor. Going forward, we also did the sterilization procedure considering all the things that I mentioned before. So for instance, we needed to use a sterilization procedure that was proper for biopolymers. We couldn't use any high temperature sterilization procedure, and also a sterilization that was proper for the in vitro test system. For sterilization, we did 2 types of tests. One that was the pathogen estimation on the scaffolds, and the other one was the dose distribution in a sample chamber, because we chose to use 25 kilograys of gamma rays for sterilization, and therefore we wanted to see the dose distribution. And finally, once all of these 3 things were designed, fabricated, and tested, we did the validation, which means that we have to probe individually each of these sections to see that they all worked or not. So now I will move, as I said, into the results and discussion, and some of the details I will address through this section. Now, to begin with, I would like you to see the bone scaffold fabrication procedure that we used. We wanted to reproduce as close as possible the trabecular bone morphology. So in that sense, we took a sample from an iliac crest bone from a cow. We imaged the bone using microcomputed tomography. And then with the image stack from the microcomputed tomography, we did a reconstruction of the bone model, and this reconstruction was then printed using a stereolithography technique, or in short, SLA, using a biocompatible resin. Using this technique that I explained before, we achieved a trabecular bone sample that, as you can see in this result, replicates the structure of native trabecular bone. In the SEM pictures, you can see the internal morphology of the trabecular bone and also of the bone scaffold. As you can see, we have similar porosity in both of the samples. Well, this is trabecular bone, but in our sample, high porosity was achieved. This was an important result because high porosity works to increase surface area. And if we have high surface area, then we can have more cells in covering the scaffold. And also when it's going to be used as an implant, porosity also indicates places for vascularization. So this was a good result. However, as you can see, the trabecular thickness was a bit higher for our scaffold, but this was due to the printing technique that we used, the resolution that we couldn't achieve a one-on-one scaffold reproduction. But I think it's okay, because you can see there is a variety in the trabecular thickness. Not all of them are the same. These two characteristics, porosity and trabecular thickness, are very important in bone because they determine their mechanical properties. Now I will go into the mechanical characterization of the scaffolds. On screen, you can see how the configuration of the mechanical testing was made. We used a procedure, a three-step procedure that was adapted from literature because we thought this way it was a more reliable way to calculate the mechanical, the Young modulus or the elasticity. When we used this procedure, we got the following result, in which we can see an elastic section for the scaffold and also an elastic section for the trabecular bone. For these two results, we obtained the elasticity and also the other two mechanical parameters, which are the yield stress and the ultimate compressive stress. The elasticity of both scaffolds are very similar, and also they are according of what we can find in literature. So also this was a very good result for us. Also, we did the dynamic compressive test because we wanted to see how the scaffolds behaved under cyclic loading. We chose to use a step loading procedure in which we increase the loading each 5,000 cycles, because this allowed us to test more samples. And this here you can see the results for bone and for this scaffold that we fabricated. This is the mean strain as a function of the cycle number. The main thing that I want to point out here is that the mean strain per each cycle remains quite similar, which shows us that the scaffold doesn't have a lot of deformation accumulation, let's say. When the strain increases sharply here is when the load also increased during the test. Now we also calculated the hysteresis curve for the samples, and in the hysteresis curve you can see if there was deformation accumulation on the sample, if there is a let's say a gap over here on the compressive strain, but this didn't happen for our SLA scaffold, so it also endured this loading, these types of loading. And you can see here the one that we got for bone. Again, the stress there is an increase in the stress, and that's because also we attributed that to the trabecular thickness that was found during fabrication. Now, for this type of scaffold, we also did a microdeformation study in which we used microcomputed tomography and digital volume correlation technique, or short DVC. I will explain this technique briefly in a two-dimensional array. However, this technique is used in 3 dimensions. Now, for this technique, we use an equipment as shown in this figure. Over here is where X-rays are emitted. Now, X-rays, high energy X-rays, go through the sample into a detector, and these are captured according to the density of the sample. So, as an example, here is an image of how, a cross-sectional image of how an image is acquired. The light gray areas are where the scaffold is, and the dark gray areas is where the air was. Remember, this is a technique that is used 3 dimensionals, but here I'm only showing 2 dimensions. So after a moment, or after a while, we can deform the volume, and as you can see, there is a difference between the first volume and the second volume. So by using an algorithm and a computer software, it is possible to divide the volume in small subvolumes, and then doing a tracking of the grayscale between one another, and this way we can obtain a deformation vector field, which gives us how the sample behave under this loading stress. And moreover, the program is able to do a strain field. Ay, perdón. Voy a volverme aquí. And the software then it's able to do a strain field distribution of within the volume. For this research, we focused in studying the normal strain distribution, which is the one that goes in the direction of the loading. Now, as the results for this, we have first the stress strain deformation curve for bone. We had to take images at certain time points. So we began without any compression, then one in the elastic section, one near the yield strength, and one near the ultimate compressive stress. And doing this procedure that I just previously explained with DVC, we were able to obtain the strain map distribution over the reconstructed volume for the bone. So for instance, in this image here shows the resulting strain map on the elastic section of the bone. For our bone, this is close to 10,000 strains, which is close to the yield point of bone, but the other sections don't show any strain yet. However, when we increased, as you can see, the strain map is focused in the middle section of the bone. We did the same study for the SLA scaffold. And again, we acquired the information at different loading points, and we have the strain map for the SLA scaffold. Again, the strain focuses barely in the middle of the sample, and at least in an inclined plane also. 2 main things I want to point out for this result is that the SLA scaffold is very similar to the bone scaffold. That was something that we wanted to achieve. And also, we wanted to see or study where strain was going to be accumulated, because remember, this is going to be used for bone cells growth under mechanical stimulation. In literature, it is found that cells tend to focus in the areas where the strain is higher, and this is where also the mechanical signals are higher. So it was a a nice result for us. Now I will move into the cell viability of the bone scaffold, because as I said, we needed to test cell viability. So we did some growing of bone cells on some pieces of the bone scaffold and tested the viability, which means how many cells were present after a period of time. And we compare with a control that were cells that were growing without any scaffold. As you can see, both of them behave in a similar way. That is a nice result because it means that the scaffold doesn't have a negative effect on the cells. And just as another result, I wanted to show you an image of the whole volume of the scaffold. You can see a slight purple coloration on the top that indicates the presence of cells. Now I will move into another type of scaffold. Remember, I said I was dealing with two types of scaffolds, and this is independent from the previous one. So we have to change our mind, let's say. So for the microfilament scaffold fabrication, this is a different, we want to imitate a different tissue. So we need a different type of fabrication technique and a different polymer. In this case, we wanted to imitate a skeletal muscle, which is made as parallel grouped fibers. So for that, we reproduced that configuration by using PCL pellets extruded with a 1 millimeter die and collected in a spooler. Then we did some characterizations of the surface of the sample, and also we saw that they could be arranged parallel to each other. The average diameter of the microfilaments that we obtained was 90 micrometers. Now we did also the mechanical characterization of the microfilament scaffold. For that, we used a configuration as shown on the screen. This configuration allowed us to test the group properties of the microfilaments opposite to the individual properties. And this was also a very good grip system that we designed, because skeletal muscle in nature behaves as a group. We got the stress strain curve and also the cyclic deformation for the microfilaments. We can see it has a elastic section and a plastic section as well, and a bit higher elastic moduli. This is within the range of cartilage, so at least we were on the same region as skeletal muscle. And quickly, for the dynamic testing of the microfilaments, we did a test in which we loaded the sample with different stress values for 3 days. This was a very long test, but we chose 3 days because muscular cells take this long to reproduce and differentiate. This indicates that the scaffold keeps its integrity throughout the duration of the test. And also for the cell viability of the microfilament scaffolds, we measured the amount of cells through time, and we saw that they increased, so the scaffold was not toxic. Here in the middle, I wanted to show you two images, one where the cells were properly attached on the surface of the microfilament, and one where the cells fell down. So this is a result that we don't want, but however, we were able to achieve the good result. And this one over here on your far right was a filament that we left for 15 days to grow with cells. And as you can see, cells started to mix between each other and doing this tissue between the filaments. Now for the second topic that we have to address is the design and the fabrication of the mechanical simulation system. We did two types of grips systems, one for the trabecular bone and one for the microfilament scaffold, each for the requirement of the loading of the scaffold. Also, we fabricated a bioreactor or sample chamber that allowed us to do the growing of the scaffold inside. It also had some inlet and outlet ports on each side, and the top grip that is shown on the top can move freely to transmit mechanical loadings. To be able to control the temperature, we used a water bath that surrounds the bioreactor. So in this next slide, I can show you the assembled system in the universal testing machine. So from top to bottom, you can see where the holding grip of the universal testing machine, then the load cell, you find the water bath that surrounds the bioreactor. Then there is a pump that moves the water into the water bath, and the blue tubings there are for carbon dioxide circulation, because remember that bioreactors have to control environmental physiological conditions. So for that, we designed and used a CO2 circulation system that is connected to the in vitro test system. So this is our system that I just previously showed, and everything that surrounds it is the diagram that shows the connection of the sensors that control the carbon dioxide. This over here is the interface that we use to, the interface that we use to input the amount of carbon dioxide or temperature that we want to achieve. And we follow these lines to see if it's working properly. Now I will go into the gamma sterilization technique. As I said, we wanted to be sure that the sterilization technique that we chose was proper or working properly for our scaffolds. We used this equipment that is available here, and we chose to use 25 kilograms of gamma rays. First, we did a pathogen estimation, which means that we counted if there were pathogens present on the samples after irradiation, and results show that, for example, in this image, that the thioglycolate is clear of pathogens after irradiation. And we did that for several samples and also several types of organisms. And as you can see, before and after, they were all clear. So we're somehow happy that, or certain that, our samples were not contaminated. And in addition, this equipment has a sample chamber in which we locate our sample inside a bag. We weren't sure, or we wanted to be sure, if the dose distribution on the sample was working properly. So for that, we did a dose distribution study in which we calculated the uniform coefficient, which is the first one, the first line. The uniform coefficient, if it's very close to 1, it means that the dose is uniform within the sample chamber. So that means that we can place several samples inside the sample chamber and they all will be receiving the same amount of gamma rays. And also we calculated, the range of dose with the uncertainty. And as you can see, 25 kilograms, that is the one that we want to use. It's right between the lower and upper values. Now I will go into the last part of the results, which is the validation. For the validation, we only did the study using the microfilament scaffold. So the in vitro test system was probed with the microfilament scaffold and not with the bone scaffold. So I will begin by showing you the results of the environmental parameter system circulation. We did 5 tests during 24 hours. And as you can see, temperature remains very close to the value that we wanted to achieve, that was 37 degrees. And it also happens with the carbon dioxide, which we did 5 tests, and through the 5 tests, the system was always achieving 5% carbon dioxide. These both conditions are required for cell growth. Now, as for the validation of the biomechanical study system, first, we wanted to validate the system by itself. And by this, I mean, we didn't use the scaffold because the scaffolds are tricky. We used a microscope slide that is a known substrate. And we didn't do any mechanical loading. So for this test, what we did was we cell seeded a known substrate, microscope slide, and placed it into the mechanical stimulation system by itself. Here you can see it in this picture. And at the same time, we had a microscope slide on the same conditions in a commercial incubator. So what we were expecting was to see if there was any difference in our system that we built and the commercial incubator. There should be none, right? And fortunately for us, that was the result. When we measured the cell viability of the cells on glass, we noticed there was no difference between each other. I mean, you can see slight difference there, but that's normal for cells. And this result is very important because it means that the in vitro test system is working as a bioreactor. It's providing the environment for cell growth on a substrate that we wanted to do. And in addition, since we were working on a self-substrate, we took some pictures of the cells. So in the lower end, we have the cells in the in vitro system, and on the top in a commercial incubator. And there is also no difference between each other. Cells are healthy, they have a star shape, and through time, they start to increase. The same result as shown on the cell viability study. So at least up to here, the bioreactor was not toxic, was properly sterilized, and also had proper temperature and proper carbon dioxide circulation. Once we had done that, we went to test the scaffold. So now we were sure that bioreactor was working. So now we went into testing the scaffold. So we did a cell seeding procedure on the scaffold. Then we did the preparation of the bioreactor and did the whole assembly together. And again, fortunately for us, the cells that were growing inside the study system are the same as the ones that were growing on a commercial incubator. So once again, the bioreactor is working properly. And moreover, this result shows that the scaffold is working properly as well. In these images, I'm sorry, I didn't say the blue dots are the stained nuclei and the green is the cytoplasm of the cells, and you can see them on the surface. Now, in addition, we wanted to do one final test, and for that, we decided to do a loading on the scaffold, because this is what the bioreactor should do, but the system should do loading. So we selected a loading function that we found in literature, and also we had a very good result because cells remain on the filament and withstand the loading. And now we are trying to do some interesting to get interesting results as cell counting and the nuclei distribution on the samples. So with that, I would like to show you the project's contributions. At least for me, this was a very huge project. There is the PhD dissertation that was written and my lovely jury here have already read. And also We submitted a patent to the here in Costa Rica. It's still under revision. Then we have 3 accepted manuscripts as of now, and also that you can read online. And also we have 2 other that we're currently working on, and one is has the results of the trabecular bone, or the trabecular bone results that we're working with Professor Gianluca. And then as a conclusion, I would like to say that, in short, we achieved our specific objectives. We did the scaffold and we characterized them. We tested the sterilization procedure, and we also did the in vitro system. It was designed, fabricated, and implemented. And also wrapping this up with the things that I mentioned at the beginning, we sort of did the cell-seeded scaffold that I was mentioning, and these results can be used as groundwork for making a study model eventually, and also to continue with towards achieving a proper cell-seeded artificial implant. As for future work, there's a lot of things that can be done. We want to test with the bone, with the trabecular bone sample. We want to test different mechanical simulation patterns. Also, it would be interesting to change physiological conditions, and we'll also do some studies on drugs and injuries on the scaffold. Of course, there are some recommendations that we have to improve, that we learn on the way. For instance, the cell-seeding procedure was quite challenging, and also we have to include a protocol for replicates, because these are very important in biological processes. Now, I would like to acknowledge the work of a lot of people that were involved in this project. The people that are here today, the people that are in the picture, I really appreciate everything, all the collaboration. And also, or with that, I say thank you to each of you for being here today. Yes. Thank you, Laura, for the great presentation. We appreciate your presentation.
For the examination, I'm just going to present the committee members after the all public leave the room. The committee member is or are integrated for Dr. Teodolito Guillén, Dr. Ricardo Starber, Dr. Leonardo Lesser, Dr. Gianluca Totti, who is in the media online, and my person, Carmen Hadris Quiroga. Now I also thank all the public that joined us with presentation and has to leave the room to start the second part. Thank you so much.
Sí, gracias a las personas integradas en la transmisión, y vamos a cortar la transmisión ahora.